Hi everyone. I'll take off my mask. My name is Emma Speh. I'm a neuroimaging engineer at Washington University. Today I'll be talking about data preprocessing and temporal analyses for NIRS data. So the learning objectives for this section will be to evaluate data quality and preprocess NIRS data, understand the importance of superficial signal regression in eliminating physiological noise, and to analyze measurements at different source detector distances. So first, just an overview of different modalities in optical imaging to get to what modality we'll be using today for this talk. So starting with near-infrared spectroscopy, or NIRS, as was discussed in previous sessions, we're able to get information about the oxygenated and deoxygenated hemoglobin concentrations in the brain. And then there's also multi-channel NIRS, where we're able to use multiple channels of near-infrared spectroscopy to gain more information about the brain. There's also diffuse optical topography, where we're able to create 2D reconstructions of the brain surface, or the underlying brain activity on the surface. And then there's also diffuse optical tomography and high-density diffuse optical tomography, which utilizes denser grids to obtain greater image quality. And then these are just some DOT studies that you can look into that go more in depth about how DOT works. So on the bottom left shows a sensitivity profile of one source and one detector used for DOT, showing our sensitivity to the scalp and brain activity. And then on the right, the flickering checkerboard on the left is the stimulus paradigm that I'll be going over with preprocessing in this talk. And the right is the DOT results in the visual cortex from a participant using this stimulus. So just a brief overview of diffuse optical tomography. In diffuse optical tomography, DOT, we use arrays of optical sources and detectors to perform functional neuroimaging experiments on task-based and resting state modes of human brain function. So our analyses of this data are broken into several pipelines, starting with modeling of the tissue and optical geometry, modeling of the light emission, diffusion, and detection through the head. And for this talk, we'll be talking about the preprocessing of the raw source detector measurements. And then there's reconstruction and spectroscopy of the preprocessed data, as will be talked by Adam after me. And then finally, post-processing analysis of those results. So the stimulus paradigm used here is the same one shown before, but it was taken on one subject using 8 blocks of the counterclockwise rotating reversed flickering checkerboards at 8 Hertz. And it's rotated 36 times for the participant. So on the top right, you can see the time course of activations in a single voxel in the subject using the continuous clockwise paradigm, otherwise CCW. So here you can see the responses in response to 8 repetitions of the wedge stimulus. And then the Fourier spectrum shown below shows a peak at the stimulation frequency of 0.0278 Hertz. And then here you can also note that there's little background noise indicating that this was some good data that was taken on the subject. So this talk will follow the NeuroDot preprocessing script as well as the Getting Started script, which is the same script written in Python. These are available on NITRC or on GitHub if you wish to download them. I'll also go more into that later in the talk. But you will here process and visualize raw source detector measurements, examine the different nearest neighbor measurements with different source detector distances, examine the measurement spectrum, explore effects of different filtering cutoff frequencies and parameters, as well as examining the effects of superficial signal regression. And we'll again use the data from the previous Renatopy experiment. And on the right shows the overall pipeline structure, starting with the DOT acquisition, going all the way down to our last step, which will be block averaging and the preprocessing. So just an overview of preprocessing. It includes filtering, regression of the superficial signal, and all of this is in hopes of reducing the noise due to physiological components of the signals. So on the left is the data shown before any preprocessing is done, where the top is a time trace of all of the signals, the center is an image view of the signals, and the bottom is the Fourier spectrum of the same data. And then the center shows the effects after, and then after block averaging all of the stimulus events in the paradigm. So first, before we do pre-processing, we look at a few data quality metrics to determine the quality of our data. So first, these time traces show our data from 750 and 850 nanometer wavelengths. And the top panels show all of the measurements we have, and then the bottom panels only show the measurements that pass through a threshold of a standard deviation of 7.5%. And then the vertical lines that appear on the bottom plot are the event points in the stimulus where the checkerboard was flashed for the participant. So then I also wanted to illustrate throughout this talk, I have figures in MATLAB and in Python using the same toolbox. So this is the same figure generated in Python. Next, with the data quality, we look at the light levels across the cap. In the top row here is just the average light levels across the DOT cap, where the lighter colors on the figure, the whites and yellows, indicate high light levels, indicating good coupling to the scalp, and then the center row is the signal-to-noise ratio for the entire cap. We also look at measurements at different distances, so the left is the shorter distance measurements, and the right is the medium distance measurements. And here we can see that this data is of relatively good quality, since there is some evenness across the cap and generally a high signal-to-noise ratio. Again, I show this figure in Python, so it can also be generated. And then I have an example here of a noisy data taken from a participant. So this was on a hearing words task, so basic audio stimulus. And then just a few indicators that tell us that we have poor data quality are the lack of consistency across the cap. So there's kind of every color you can see in this middle row, as well as on the top row. And then the maximum signal-to-noise ratio here shows to be about 1 or 2, whereas we look for something more closer to 20, hopefully greater than 20 for good data. And possible causes of this are due to a poor cap fit on the participant. Most likely it's due to the hair interfering with the optodes on the head. So next, another way we can look at the data quality metrics is by analyzing the signal and the noise. In the top left figure is a zoomed-in view of the time traces. And here you can see a pulsatile response to the participant's pulse, around 1 hertz. And then on the bottom left is a spectrum of the same time traces where you can see a peak around 1 hertz for the pulse, and then we are looking for another peak around the stimulus frequency, which is about 0.0278 hertz. And then on the top right is a log linear plot of the light fall off through the head. So this here shows expected diffusion of the light through the head. And in the bottom right histogram shows the amount of measurements that fall above the threshold of 7.5% that are cut out. Again, the same figure in Python. And then a few metrics that we look at to see if the data is good or not. In the bottom left, you can see on this noisy data, you can't find much of a peak for the pulse. So that tells us that maybe the cap isn't on the participant's head properly. On the top left, you can see some pulsatility in the waveforms, but there's less consistency across all of the signals. And then in the top right figure, just the large spread of the sources and detectors shows that the coupling was not consistent across the cap. So to begin the pre-processing after we've looked at the raw data quality, the first step is taking the log mean and finding the clean measurements, which would be the measurements that fall above the threshold or below the threshold of the standard deviation being less than 0.075. So on the left is the data before we've done any processing. On the right is just after we've taken the log mean and the clean measurements. Here you can see some slight improvement in seeing the peak of the pulse and the stimulus, but not much change has happened yet. And in the center plot, you can start to see some periodic response due to the stimulus, which we know is about 0.0278 hertz. And yeah, you can see the pulse and stimulus frequencies in the average spectrum on the bottom row. And next, I just have a comparison of the MATLAB and Python versions of this. And then the next thing that we do is we view measurements from different distances to help us with our interpretation. So the goal of looking at this is that at shorter separations, we can reveal systemic signals such as the pulse and respirations signals, and then at longer separations, we can find the responses due to the underlying brain function. So at this step, we're not able to see any information about the brain function, but in the shorter distance measurements on the top row, you can see the pulse frequency around 1 hertz. And then again, I show those figures in MATLAB and in Python. So then the next step in preprocessing is detrending and using a high-pass filter. So by detrending the data, we remove a linear trend or a drift from each measurement. And then the data are high-pass filtered at a frequency of 0.02 hertz, where everything below is attenuated and everything above will pass through the filter as shown on the bottom for a spectrum. So here we're just narrowing it in on the information we are interested in looking at. And then you can see still in both figures, we can still have the stimulus frequency and the cardiac pulse. Future steps, we'll try to eliminate the cardiac pulse from our data. And then again in Python. And then by viewing the filter measurements at different source detector distances, we can see some slight changes in the short distance measurements, but still at this stage, there's not much that can be gleaned from the long distance measurements. And then we can also do this step in Python. So the next step in the preprocessing is a low-pass filter. So we use a cutoff frequency of 1 hertz to filter this data. And with this, we cut out the pulse frequency, as you can see in the bottom Fourier spectrum. So the time traces, if you look at the top row, they show less high-frequency noise after this step, and the image of the measurements in the center shows a more clear periodic response to the stimulus. And again, now you can see there's no longer the presence of the cardiac pulse in the bottom right. And then the same results from Python. And then the filter measurements at different distances now show reduced high-frequency noise at all of the source detector distances. So the zoomed-in view then shows that the 1 hertz cardiac pulse was filtered out as we intended. So the next step is superficial signal regression, and with this step, we're hoping to filter out any noise induced by the physiology. So an estimate of the superficial signal is taken, and we calculate the mean of the first nearest neighbor measurements. So all of the more shallow measurements to regress out the information that's due to the scalp tissue and the skull, which here is about 13 millimeters in depth. And by regressing out the systemic signal from the time traces, we get greater spatial specificity. And the figure on the right shows our sensitivity, where it's very sensitive to those measurements. Here is a figure showing the estimated superficial signal that will then be regressed out of the data. So the signal and its frequency spectrum show the features from the short distance measurements that we are not interested in. And once we regress this out, we'll be left with just the long distance measurements that we're interested in. If you see in the bottom figure, we just have the strong peak around the stimulus frequency of 0.0278 hertz, and little noise due to anything else. And again, and now at this point, you can see an even stronger periodic response in the top two figures. And then by looking at different source detector distances at this step, at the longer distances, you can very clearly see a periodic waveform around the frequency of the stimulus, which is, again, 0.0278 hertz. And that is the information that's actually due to the brain function that we're interested in. So after we've done superficial signal regression, we still want to denoise the data a bit more. So we apply a second low-pass filter, and we resample the data. So we use a second low-pass filter at a cutoff frequency of 0.5 hertz, and then resample to 1 hertz. So now here we have the sampling reduces the number of data by a factor of 10. And then again, this figure is generated from Python and MATLAB. So the second low-pass filter further gets rid of the high-frequency noise. And then here I show the effect of changing the cutoff frequency. So we can reduce the filter cutoff frequency to 0.05 hertz to reduce the high frequency noise, which would only leave waveforms at the stimulus frequency. But this happens at the expense of possibly altering the temporal features of the stimulus response that occur around 0.028 hertz, and then possibly altering the phase of that response, as you can see in the long distance measurements at the bottom. And then again, we can generate these figures from Python as well. And then the last step in preprocessing is block averaging, where we block average based on the stimulus synchronization points, or wherever there is an event in the stimulus paradigm. The time traces here show a clear response due to the rotating checkerboard stimulus because it's averaged over all of the times where the checkerboard was rotated, and then the measurement image shows clear spatial variation to the response, as we would expect as the wedge rotates through the visual field. And then when we look at different source detector distances at this step, we can see that the medium and long distance measurements are nearly containing all of the strong stimulus response with very clear wave-like structure at the frequency that we expect for the stimulus. So in summary, we evaluated data quality and preprocessed data using the NeuroDot toolbox, and we used visualization of multiple metrics of data quality, including light level falloff, coupling coefficients, and the pulse signal-to-noise ratio. We went over the importance of superficial signal regression to remove the systemic physiology and make the spatial specificity to the brain function. And then lastly, we analyzed the measurements from different distances, where the closer nearest neighbor measurements provided the systemic superficial hemodynamics, whereas the deeper measurements provided greater brain specificity. And lastly, by having multi-distance overlapping measurements, as we do with DOT, that will be realized with image reconstruction, as will be talked about by Adam after me. And I also want to highlight as sort of the interactive part of this talk. You can use cloud-based processing using XNAT. So I have a brief walkthrough of how to register for an account on XNAT if you want to use it. This is also posted on the NITRC page for NeuroDot with a walkthrough of how to use it. So after this, you'd be able to run the pre-processing analysis on a set of our 10 data samples, including both the retinotopic mapping of the visual cortex, as shown in this presentation, as well as mapping of hierarchical language processing. And then from there, you're also able to view and download results from pre-processed HD-DOT data. So XNAT is a cloud-based processing service that provides a container service that can be used to run scripts, such as in MATLAB and Python, on many data sets. Since DOT processing can be very highly computational, it requires a lot of resources, including possibly the MATLAB license, which may be a limitation for many people. XNAT provides a platform which provides the resources to run those processes, for example, the NeuroDot preprocessing pipeline. So if you go to the NITRC page, there will be a document that guides you through registration for an account, and this is how you would register for an account. And then from there, you would have a project created, which you can then use the NeuroDot preprocessing pipeline on that you would need to enable. And then from there, we have a list of 10 subjects, including just some basic stimulus paradigms that we use in our lab, including the retinotopy, auditory tasks, as well as a noisy data set that you can use to manipulate different parameters to try to eliminate the noise. So from here, you can also adjust the parameters that you want to use. So this container will just run your code, the data on our code, and generate all of the results. But you can change the parameters if you wish to just see what the effects of different preprocessing steps are. So there's a file that you can download, and these are our preprocessing parameters in NeuroDot. And just if you want to mess around with it, this is an explanation of what all of those do, where bthresh is the threshold that we set, which is at 0.075. There's a set of logical flags if you wish to skip a particular step in preprocessing to see the effect that has. You can also set the cutoff frequencies for each of the filters, as well as the frequency for resampling. as well as the tolerance for resampling as well. And then the DT and TP are information about the stimulus paradigm that's used. So there you can run the container once you've adjusted the parameters and then view the results. And in the results folder on XNAT, you can download all of the images that are generated, as well as the completed output notebook that if you have Python or Jupyter installed, you can open on your own system. So an overview of the different tools you can use to do pre-processing. So the Neuro.toolbox is available on NITRC and MATLAB and is in development in Python, currently also available on NITRC. And the other pipelines, such as reconstruction, head modeling, are going to be available on NITRC later this summer. We also want to highlight that Jupyter Notebook is a very useful tool for toolboxes like this to train people and to get more people using them and to learn how to use data processing. So Jupyter Notebook is a really important tool, and then XNAT is just the cloud-based platform, again, that provides the resources for container-based processing, and the hope is that this will facilitate some standardization across the field, and it removes the limitations for running high-performance computational tasks and requirements for locally installed software, like MATLAB in particular. So in summary, the three main objectives from before, for evaluating data quality and to pre-process data with the Neuro.toolbox to understand the importance of the superficial signal regression and analyze measurements from different distances. And now also, you should be able to run the pre-processing analyses on the set of 10 data samples. And then the Neuro.preprocessing script, along with that, you can use XNAT to produce the results. And I just want to acknowledge the team that developed Neuro.and XNAT as well. And that's all.